Okay, hello everyone. Good afternoon. It's time for the International Barley Hub seminar, and we've got a seminar on roots, which is one of my favourite subjects. So, just to remind you that the seminar's been recorded, and that if you want to ask any questions, then please put them in the chat as we go along. But without further ado, I'll pass over to Pavla Navratilova, who's going to present our speaker for today. So, over to you, Pavla.
Hi, hello. So today's speaker is Nikola Korinkova. She's from Crop Engineering and Biotechnology Group in the Catherine Institute in Olomouc. She does her PhD under supervision of Véronique Bergogneau. And they work as a group, focuses on functional genomics of barley with a particular emphasis on root system development. They study the interplay of phytohormones and genes, and to study the genes, they use targeted gene editing, and I think Nikola will also tell you about gene regulation in this process. So, Nikola, yeah, the floor is yours.
Thank you very much for the introduction. I'm very happy that I have this opportunity to present you our research, and I will tell you more about the research on the crown root formation in barley today. So a little bit more about our group. We are located in the east of Czech Republic in Olomouc. Our research institute, Catherine, belongs to the Palacký University in Olomouc. And for several years already, our group is focusing on barley research. We are actually divided in two smaller groups. One is more interested in molecular farming and using barley for molecular farming, especially for production of recombinant peptides in barley. And the second subgroup is more interested in exploring the mechanisms of tolerance to abiotic stress and how we can use it for crop improvement. A lot of our activities are connected to barley transformation, and we are very happy that we have also a very close collaboration with the Usosko company, who has the accredited GMO field where we can grow GMO barley or NGT barley and conduct field trials as well. Why do we focus on roots? They are very important organs for plant life. They are responsible for water and nutrient uptake and the interaction with the soil, including the microbiome of soil. And in recent years, we can see a lot of interest and growing interest in the research of roots, especially in crops. And I think that we also believe that it has a lot of potential because the overall root system architecture of plants shapes how the plants will tolerate and how they will respond to stress. And I think that the mechanisms of root growth plasticity in plants hold also a lot of potential for how we can make crops that will adapt to challenging environments better. Improving the root traits could have a big impact on crop productivity under various environments. In our group, we specifically focus on the research of crown root formation. Crown roots are a specific type of adventitious roots in cereals. They originate in the stem base, and in the adult plants, they form the major part of the root system. They further branch to explore a bigger volume of the soil and to acquire more resources. So they are very important for the plant life. We are especially interested in studying the genes which are involved in crown root formation. And this is also the point where I will focus in this presentation, but my colleagues also are interested in the study of the organogenesis, in using classical histological approaches to describe the formation of the organ. And we also study the local auxin maxima, how they are created and how they affect the initiation of the crown root primordium. And we are also interested in studying the pathways that are already described in model species and whether they are whether they have function also in barley or whether they diversified in function. So our group has already published last year a dataset related to crown root development. This was an initial dataset where we produced transcriptomic data relating the where we followed the differences in the transcriptome between the first day after germination and 10 days after germination. And it was useful for studying the overall changes which are connected to the development of the stem base and including the crown root primordia. But it also led to identification of certain challenges which are connected to the study of crown root development. That is the fact that when you are collecting the stem base, you're collecting it with probably several crown root primordia already present. However, each of the crown root primordia can be in a different developmental stage. And that is a problem for studying the genes which are involved in certain developmental stage. So we continued and improved our experimental setup, and we actually adapted the methodology that was already known for lateral root initiation in Arabidopsis. The lateral root inducible system, which is already well known and established in Arabidopsis and other model species, and it was useful for identification of genes which are involved in this biological process. So we adapted this protocol to crown roots in barley. It is based on the fact that polar auxin transport is the major factor in determining the crown root formation and also the formation of the local auxin maxima. So actually in this protocol, we inhibit polar auxin transport for the first 8 days after germination. And then at one point, we treat the seedlings with auxin. And we then collect samples at certain time points after the treatment. So the first indicator that this system is working was when we checked the transcriptomic data for the marker of crown root initiation, and it is the crown rootless 1 gene, CRL1. And we could see a strong upregulation of this gene already 1 hour after auxin treatment, while when we used the previous data set from the first day to 10 days after germination, it was not differentially expressed in that data set. So that was great, but then we also had to determine which time points after auxin treatment will be useful for us and useful for especially for studying the initiation, so the first step of the organogenesis in this case. So we did a histological study where we first collected the stem base before treatment with auxin, and then after 24 hours, we still did not see any visible changes on the cross-section. But when we collected the stem-based cross-section after 36 hours, we could already see a small crown root primordia present. That is actually quite progressed stage of the primordia development, so that was quite late for us. That's why we focused on the first 24 hours to really target the initiation events. And then from these samples, we produced RNA-seq and ATAC-seq datasets that I will describe in more detail next. So in the RNA-seq data, we follow the time kinetics of 3, 6, 9, and 24 hours. And we observe dynamic changes in the first 24 hours after auxin stimulation in both the upregulated and the downregulated genes. Already at 3 hours, we can see very major recruitment of transcriptional regulators, as well as factors important for shoot and root development, and many genes related to hormone level regulation. At 6 hours, it's quite interesting. It's quite early, but already at 6 hours, we can see changes in cellulose catabolism that could be related to the preparation of the tissue for cell divisions, and also for accommodation of the future crown root primordium in the stem base. And interestingly, at 24 hours, we could see a lot of genes related to cytokinin biogenesis, and I will also mention that a little bit later. So first thing that we checked was, of course, the level of conservation of the pathways related to crown root initiation. So in rice, there's already many years of research, and the pathways leading to crown root initiation already well established. So actually also in our data, we could see many genes which are putative orthologs of those regulators in rice. And in our data, we could follow the activation of those genes at certain time points. At 3 hours, it was those genes especially which are involved in the first decision of the initiation, the onset of the first cell divisions, and then also regulators of root apical meristem specification and maintenance. And later, after that, we could also observe activation or changes in gene expression of genes which are actually already described in rice to be downstream genes of the first line regulators. So it was nice that we could confirm that also in barley, and also in later points we could see more genes related to the auxin cytokinin crosstalk. But to not look only on the things that are already described in rice, and to try to look at some novelty. Interestingly, we found among the top upregulated genes in our datasets, we found a lot of genes related to phytohormone levels, and especially, of course, it was auxin, the most represented one. But apart from auxin and cytokinin, also strigolactone metabolism, ethylene, jasmonic acid, and gibberellic acid. The gibberellic acid, the metabolism and the gene responsible for that was actually one of the most upregulated genes in our dataset in all of the time points. So there was very persistent reaction of this gene. And we also, apart from the hormone metabolism, also many genes involved in the phytohormone signaling. When we can also find among those top upregulated genes already those that were described before in rice, but also some new ones. For example, the LBD27 in barley. Its ortholog was not described to have a role in crown root initiation. However, here it appears really high on our list of the top upregulated genes. So it's really interesting, and we could definitely look at this gene further. And yeah. And then also what was interesting, we could see differential expression of many genes related to chromatin remodeling. That was also something that was already suggested in rice. However, it was not studied in more detail after that. So this is something that we addressed with producing and analyzing our dataset of ataxic data. So ataxic data will give us the information about the accessible chromatin regions, and we produce those data for the same time points as for the RNA-seq. So we had idea about the chromatin accessibility changes and the transcriptional changes for the same time points. However, for this dataset, for the ataxic dataset, we could see way lower dynamics, I would say. A lot of the sites and majority of the sites were actually shared between all the time points, and that can be actually related to the fact that we are still in the same tissue, which is only like couple of hours from auxin treatment, and the differences in the individual time points are not maybe so high because of that. And when we looked on the distribution of the accessible chromatin regions relative to gene features, we could see very typical distribution with more than 60% of those sites in distal intergenic regions. And what was good indicator of good quality of the data was also a relatively high percentage of actually the sites in promoters. That was good, and in 3' UTR. This is typical for, and it was already described for the genome of the size of barley. We also checked the correlation between the data between ataxic and RNA-seq. So whether increase in ataxic data actually correlates also with the increase in gene expression, which we indeed observed. But what was more interesting for us was to look at the sites where the accessibility in chromatin is changing between the state before treatment and after treatment. And so here also the dynamics was not so high. The differences were not so high, but I mean, here maybe the response in the chromatin accessibility is not really so quantitative, not as much as in the transcriptome, but we can see very relevant sites. So maybe it's more like qualitative changes in the chromatin state. However, most of these differentially accessible regions were located in distal intergenic regions. So for that, we cannot really discuss on their function because when you do a simple annotation of those sites, the program will only annotate them to the closest gene. But if it's hundreds of kilobase or hundreds of thousands of kilobase away from the gene, it's probably not relevant, or it's something that we cannot really address without further analysis. Then when you look on this graph, actually what we focused on was quite small percentage of the data. So there's still a lot of unexplored things. It was very interesting for us to look at the specific sites in the genome and the genes that were in close proximity to those differentially accessible regions, and we could see very important regulators of the crown root formation, putative orthologs of those that are described in rice, especially, and so we can see that the process of the changes in the chromatin accessibility are very important for their recruitment, and it gave us another idea of the regulation of this process. But to further focus on some specific transcription factors, we wanted to select some interesting candidates. This selection of the candidates was consulted with Professor Pascal Gante, who is a specialist on crown root development in rice. And together with him, we selected 2 transcription factors, which were described to be to belong to the CRL1 network in rice. So as I said, CRL1 is key regulator of the process in rice. So its network is also very interesting to address. But however, there were not a lot of details known about those 2 transcription factors, and basically the only information about them was about their expression. That was it. So we wanted to continue with the characterization of these 2 transcription factors. And so the first step was the identification of the orthologs in barley. With the ortholog of HvNAC013, it was relatively easy because there was only one ortholog, a possible putative ortholog in barley. However, the situation was a little bit more complicated with the YRF28, and I will also come to this a little bit later. So when we check the expression of those 2 genes in our system, in the crown root inducible system, we actually see that the NAC gene is not somehow changing the expression. It's stably expressed in the stem base. However, in the previous dataset, we could see that it was upregulated in the later developmental stage. So maybe it's just it has different roles in different stages of the development. And the CBF12C actually very specifically responds to auxin treatment, and its upregulation is temporal between 3 and 6 hours, and then it drops again. To characterize these 2 transcription factors in more detail, we generated DAP-seq datasets for both of them. So DAP-seq will give us idea about all possible binding sites of those transcription factors in barley genome. And here we actually utilize the datasets that I already presented, the ATAC-seq, for searching for the binding sites of NAC13, which are possibly in the accessible regions, and to also look how this relates to the differential expression of genes. And originally, we started with really huge numbers of possible binding sites, but when we filtered out the distal intergenic regions, and when we specifically focus on the differentially expressed genes, we got to 238 binding sites. So that's already a number that you can work with more easily. And when we checked those binding sites that were in promoter regions of genes and connected to upregulation, we could find between them CBF12C, so the other candidate that we studied. So that was very interesting and very fast. We could already find this interesting link between them, and we could already like think if they really work in the same pathway. But among the other HvNAC targets, we also found other already known regulators of the process, or orthologs of the regulators of this process from rice, that could link HvNAC013 to crown root formation. So it was, for example, this CRL5 gene, where it was the HvNAC binding site was also found in promoter region. Then among the targets, there were also many genes related to auxin homeostasis, signaling, and so on. The lists are, of course, very long. So this is just a couple of examples. And then the same was done for the CBF12C transcription factor. And here, the number of binding sites in the genome was way, way lower. So it was a little bit easier to work with that data set. And when we looked on the sites connected to differentially expressed genes, we could find GATA, transcription gene coding for a transcription factor, which was not described before in this process, not even the orthologs. But GATA transcription factors are important for lateral root initiation in Arabidopsis. So this could be an interesting candidate to focus on further. And another CBF12C binding site was found in the superlocus of CRL1, which is very, very interesting because this superlocus, it was described 3 years back, is very conserved among many plant species. So the presence of the binding site here is really interesting for us, and we actually couldn't wish for a better genes to find among the targets of CBF12C, because CRL1 is one of the key regulators of this process. So when we found this link between CBF12C and CRL1, it was a very good indication for us that CBF12C could really be involved in this process. And is this definitely something that I think would be very interesting to address further? What is, if really this could have impact on the expression and regulation of CRL1. So I'm looking here for some links of HvNAC and CBF12C to crown root formation, but actually they belong to very typical stress-related families. CBF12C belongs to CBF3 subgroup of CBF genes, and it's part of the frost resistance H2 locus. So when I found out this, it already led me to the idea, like, what is the connection between crown root formation and these stress responses, and are they connections? I don't know. And this locus is very important for the freezing tolerance of barley, and in the evolution and in the adaptation of barley to temperate climate, this locus expanded a lot, which is also very interesting, because when you see this list of genes, they are very closely related. However, you can see that CBF12C is actually the only genes from this locus that very specifically responds to auxin treatment. And what is also really interesting is that another gene, CBF6, also responds, but in an opposite way. It is very strongly downregulated. So this is something that I think also would be nice to continue to address. And another link between these two transcription factors and the abiotic stress responses was when we checked their DNA binding motifs from the DAPSEq datasets. because for NAC013, we identified these two, the best scoring motif and the second best scoring motif, and both of them belong to the cluster 3 of NAC transcription factors, which are also specific for transcription factors involved in cold tolerance. And for CBF12C, it was the typical C-repeat CRT motif, which is also well known to have a function in low temperature, drought, and salt stress. So it's also interesting to see that both of these transcription factors actually are related to similar type of stress factors. So it's another indicator that they might work in the same pathways, not only in the crown root formation, but maybe also in the abiotic stress. And to extend a little bit more on this, we also check their expression in stress conditions, in the osmotic stress from salt treatment, and in water deficit condition, and both genes responded more specifically to salt treatment, while NAC was upregulated in this condition. CBF12C showed more trend toward downregulation. And what could be the relation to crown root formation or crown root development? In salt stress, there is decrease in the number of crown roots. So just to summarize, I think it's really nice that we found these links to crown root formation, but we would definitely need to extend on this and continue to see where exactly these transcription factors could fit in the pathways leading to crown root initiation. And do they really positively affect crown root initiation or not? And what happens after abiotic stress conditions? Is this positive impact on crown root initiation lost or not? So this is something that will be the matter of our future studies. Just to summarize, here we provide initial data sets relating to crown root initiation process in barley, which will be a very valuable resource for us for the future in helping to prioritize candidate genes for further functional characterization. I think we know that the formation of the root system architecture is a very complicated process. It's the interaction of the genetic factors and the environmental factors. So I think there is still a lot of space for discoveries, and discoveries also maybe of the kind that I showed, and more. And a lot of our work was somehow connected to the cis regulatory elements and the regulatory elements in general in the genome. So I think that there's a lot of potential in exploring their function and maybe trying to target them in the functional studies. So I would like to thank you for your attention, and I would like to thank all our collaborators and the funding. Thank you.
Thanks, Nikola. That was a really interesting talk. There are no questions in the chat just now, but hopefully people will put them in, although there comes one straight away. So, so Gwen Kirchner has asked, what could be the benefits for the plants to inhibit crown root initiation under cold stress?
I think in general, under this abiotic stresses, the plant needs to save the resources and needs to maybe relocate the resources to other parts of the plant, because it has to especially produce the grains. And so when it already has some established root system, this is probably not the priority to make more crown roots. This is my...
So another couple of questions about the physiology more than the transcriptomics elements. But so you'd imagine that the junction between the stem base and the crown root initiation zone will be affected by the depth of the seed in the soil. Is that correct, or is that genetically predetermined?
I'm not sure.
So you'd imagine that the crown roots would keep initiating until you reach the point where they're not in the soil anymore. So
Yes.
so I guess that differentiation zone must be dictated by the presence of soil or light or something like that. Is that?
Yes, well, I don't know much about that, but I definitely know that when I did, for example, experiments in hydroponics, and when I had when the stem base was above the solution, the crown roots they formed, they emerged, but they didn't grow further. So definitely there is some regulation, and the sensing of the position of the stem base in the soil. Yeah.
Cool. So I got a question from Yung Ching, which is, based on your data, is crown root initiation development conserved between barley and rice?
I would say that you know, of course, that preferentially we look on the genes which already have some described function. So if I check those genes that are described in rice, then yes, I can see a lot of conservation. If I would have to comment on the whole data set, it's quite difficult because a lot of genes in barley are not so well annotated, and yeah, so it's then to comment whether they're conserved. It's hard to say. And also comparing the whole transcriptomic data sets between rice and barley, this is not something that I did, and I think it would be quite difficult. I'm not sure.
Sure. Okay. So I have another question, which is so the genes and transcripts that you identified in the initiation zones for the crown roots, is there any similarity between those in the crown roots and those in, say, lateral root formation or any other root formation, or are they vastly different?
The gene families are very similar. Yeah, we can see. I mean, here this data set is specific for auxin response. So of course, we see the AUXIA, ARF transcription factors, and these are very typical also for lateral root initiation. So in this sense, yes. But if the same gene functions in both lateral root initiation and crown root initiation in the same species, I would say there is more specialization in the function.
Okay. Pabla?
Yeah, I want to ask about your discrepancy between the transcriptome and the open chromatin. Have you looked a little bit more in detail, especially at the cases where you would see like genes shut down, but if I understood well, you could still see the peaks, and the other way around? How many, do you have an overview of what proportion would be this like in this disagreement, like really big disagreement, I would say?
You mean when I saw a peak, but it was downregulated, or?
Yeah, or shut down even. Or did you see such cases? If I understood well, you didn't really see much correlation with the differential gene expression analysis.
Yeah, it was actually less than 50% the correlation. Yeah, but I didn't look so much into the details of this because it was not really the objective of our study. So yeah.
Yeah, so because this would mean that the genes maybe stay potentiated, kind of, or the regulatory proteins don't really. So did you consider the footprinting, ATAC-seq footprinting, to maybe reveal the changes in the occupancy of the open chromatin?
Yes, I think it would definitely extend the findings that we present here. It can be.
Yeah. The question is if you have enough data for that. If you have, because that requires higher throughput.
No. We would need to
you don't have enough.
repeat. I think we would have to repeat.
Okay.
Yeah.
Right. Nikola, I have another physiological question, I think, which is, you mentioned lots of stresses like cold and drought and things like that. Is there any evidence to suggest that the crown roots are associated with wind, for example? Because you'd imagine that they'd be quite good at bracing the plant against lodging, for example. So I just wonder if there was any suggestion that you get more crown roots in windy conditions.
That's a very interesting question, but I have no idea.
Just looking out the window here, it's very windy in Scotland, so actually, yeah, it might be quite useful to know.
I would maybe expect this for other root types, and probably, I don't know, I would maybe not expected this for barley, but maybe for maize it could be.
Sure. Yeah. Cool. Okay. Has anyone else got any more questions? And if you have, then pipe quickly, because otherwise we'll close the session very soon. We'll give everyone a last few seconds just to put any questions in, and if not, then thank you very much, Nikola. It was a really interesting talk, and hopefully lots of people will pick it up on the recording as well. So thanks, and we appreciate you speaking at the IBH seminar.
Thank you.
Bye.
Thank you for inviting me. Bye-bye.
Bye-bye.